Okay, well, let's go ahead and get started here. A couple of announcements. Quiz for quiz chapter 11 for this set of lecture slides still due on Friday. We will cover certainly all the material by Friday. Okay, so I don't think we'll get to it all today, but you'll be ready to take it on Friday. That snow day is still affecting us at this point in time. Next week at some point, and I don't know exactly when, probably Wednesday, though, I would imagine. Possibly Monday, but probably Wednesday, we will have a group work, okay? So I'm fairly certain it's going to be on Wednesday. But we'll do a group work on that day, and then next week we'll have a problem set due as well, okay? So I'll be posting that problem set either this afternoon or tomorrow, and that will probably be due towards the latter end of the week, okay? All right, so our riddle, what two words have thousands of letters in them?
Mailbox.
What? Mailbox is one word. Isn't mailbox one word? Well, you're close. Post office. Post office. Compound word, yeah, compound word. What did you say? What did you say? Oh, thou and sans. Ah, very good. Okay. All right. So last time, I'm stuck behind here for the day. My wireless system is not working. But last time we left off right here, looking at the pathologies that would result from an accumulation of LDL in the bloodstream due to this condition called familial hypercholesterolemia. I got it right again. So LDL, remember, is a small, relative to the size of a cell, fairly small lipoprotein particle, carries a fairly significant amount of cholesterol esters in it. And those can slip into, especially when levels of LDL in the bloodstream are high, can slip in between endothelial cells and get into the extracellular matrix surrounding essentially the wall of artery. In that environment, they can undergo oxidation, and that oxidation will lead to an immune response that will essentially have monocyte recruitment. That will then be, those monocytes will be converted to macrophages, and those will start to absorb fairly significant amounts of the LDL through endocytosis. They'll have the LDL receptor, and they'll take those in. In that endocytotic process, just like in the liver or any other tissue, those endosomes will fuse with a lysosome, and that will then spill fairly significant amounts of cholesterol into these macrophages. They'll keep taking that LDL in, and eventually they'll take in so much that it will become toxic to those cells. They'll undergo necrosis or apoptosis, split open, spilling essentially cholesterol and cholesterol esters into that extracellular matrix, leading to these plaques that then, of course, can cause closing of the arteries. And if that happens, of course, in the heart or the brain, that can lead to stroke or cardiac arrest. So obviously a very serious set of conditions that we're describing here. So what we want to go ahead and do today is talk a little bit about treatment of this. So let's go to here. Okay, and what you're looking at here, it's a fairly busy slide, lots going on, but really what we're thinking about is the two main sources of what might be considered intracellular cholesterol. So we'll box that in here, cholesterol in the intracellular environment, okay? As we have discussed up to this point in time, whether this be through an endogenous pathway or an exogenous pathway, LDL, well, in this case, because it's called LDL, would be an endogenous pathway. But that cholesterol can be, of course, uptaken from the cell by, cholesterol that's in the circulation in LDL. Okay, so we've talked about that. That would be receptor-mediated endocytosis. Okay. Now, the other aspect of this that we haven't really discussed, though I've mentioned it, is the fact that cholesterol itself can be made within the cell. Okay, almost all of your cell types, if not all of your cell types, have the capacity to carry out a fairly complicated but efficient metabolic pathway that will lead to cholesterol. Okay, that pathway begins here at a fairly familiar molecule, acetyl-CoA. Of course, acetyl-CoA is what pyruvate derived from glucose becomes, and of course amino acids can become acetyl-CoA, and certainly fatty acids, as we will see in our next lecture, can become acetyl-CoA. Okay, we'll look at these steps in our next lecture, but acetyl-CoA can become this molecule beta-hydroxy beta-methylglutaryl-CoA, which is, not fun to say. Okay, so it can also go by HMG-CoA. I like HMG-CoA, that's easier. Okay, we'll look at those steps. Like I said, we'll talk about ketone body protection, and this is a part of that process. But that's HMG-CoA, and at that point in time, really enter into what might be considered the cholesterol synthesis pathway proper. Okay, starting with this enzyme, HMG-CoA reductase. Okay, that will take in a single step HMG-CoA and reduce it to mevalonate. And then mevalonate through multiple steps, and I mean multiple steps, lots of them, like 20 or 30 of them. You can thank me, you can thank me later for not going over them. Okay, but a large number of steps will lead to eventually cholesterol. Okay, so those are your two main sources of cholesterol within a cell. Either take it up from circulation or make it itself. Okay. Now, given that, let me go ahead and ask you a question. And considering what we've talked about with familial hypercholesterolemia, let me ask you this question. Okay, what might be a good drug target to treat FH? Okay, FH, remember again, is that difficult thing to say. So what do you think might be a good drug target, considering all that we've talked about, for FH? Okay, so I'll give you a minute 45 to discuss that. Obviously, we are going to look at some drugs here and what they affect. Okay, on your marks, get set, go. Uh-oh, I get a connection interrupted. Responses may be delayed. Attempting to reconnect. All right, talk amongst yourself. Okay, talk to yourself. Talk to your neighbor about this. Try and fix this. Are you able to respond?
No. You don't even have the right question.
Awesome. Okay, so apparently I'm not getting Wi-Fi signal in this room. I don't know if you guys are getting Wi-Fi signal in this room, but... Huh? You are?
Wi-Fi works.
Then it's my computer. Okay, so I can't get it. All right, I'll try one more time. That's depressing to me. Keep talking amongst yourself. Okay, so I'm not going to be able to get there. So what we're going to have some people do is go ahead and shout it out. What do you think? Okay. Okay, so we got some HMG-CoA reductase. Anyone else? No?
On the other slide, glucagon was shown to like inhibit going from HMG-CoA to however you say that.
Mevalonate?
Mevalonate,
Yeah.
Maybe something like upregulating glucagon signals or
Okay.
AMPK to inhibit that metabolic pathway, or just inhibit acetyl-CoA's ability to go to HMG-CoA, so that you're depriving cells of one way to make cholesterol intracellularly.
Sure.
And then maybe another way is, I don't know if there's a way to inhibit cholesterol absorption in cells, but I think that's a lot more complicated.
Right. Right. Okay. So your idea would be, so we got HMG-CoA reductase. So someone's mentioned this here. Others have said, or what you're saying is essentially maybe upregulate some portion of this pathway, or maybe even this pathway here, right? But all of these are centered on that HMG-CoA reductase. So it turns out that, right? I mean, those are all viable drug targets. It turns out though that the one that the drug companies have successfully been able to use as a drug target to treat this disease is HMG-CoA reductase. Okay, so as you see here, we'll come back here. This molecule here, mevalonate, is the product of that HMG-CoA reductase. That molecule is this guy right here. Okay, they also, just so you have some concept, remember this is HMG-CoA. That molecule is this guy right here, okay? And several drug companies have made a family of drugs, these guys, okay, and then this guy here, okay? Common names for these, Mevacor, Pravachol, Zocor. Okay, if you'll notice these three over here, okay, all of them have a moiety on them that look very similar to either the substrate or the product of that enzyme. Same with this this drug here, this compound Lipitor, which is commonly used to treat this disease. It also has something that looks very similar to this. Okay, so the idea with this is that these drugs will inhibit, and that's exactly what they do, inhibit this step here. Okay? And inhibiting that step there, the whole concept would be to ensure that cholesterol levels inside the cell stay low. And the reason that you'd want that is because then if those cholesterol levels remain low in the cell, then the cells are more likely to take up cholesterol from the circulation. And here's the reason why, okay? When we look at this, and this is going to be an important aspect of our discussion here, cholesterol in the intracellular environment, while it certainly can become cholesterol esters packaged and then sent out of the cell, there's a byproduct of cholesterol metabolism, and that is a family of compounds called oxysterols. And oxysterols, as you see here, this is very important for our disease condition, will actually inhibit receptor-mediated endocytosis. Okay? So what the use of these drugs does is ensure that there's a small amount of oxysterols being produced in these cells so that receptor-mediated endocytosis can continue to occur. Okay? Remember, this condition is the result of defective LDL receptors. Okay, in the heterozygous sense, there's only half of the LDL receptors are functional. So to then downregulate that pathway in the cell makes this condition even worse. Okay, so that would be an important thing to do in terms of just slowing down cholesterol synthesis within the cellular environment. These drugs have demonstrated to do that and have fewer side effects than maybe some of their competitors that failed. Okay, and all of you, kind of smiling, because if you watch sports or anything on TV and they have some drug, they have some commercial for some drug. You get all these happy people and everything, and they say what great things their drug does for you, and then they go into hushed tones, and for the next minute and a half they list off all the side effects, and by the end of it, and if you feel like you can't move your legs or you're paralyzed or something, call your doctor immediately. And then they go back, and this drug will save your life, change your life. So there's always side effects, but apparently these don't have that drastic of a side effects. Okay. All right, so that's one way to treat these, and certainly individuals with this genetic disorder will take these drugs, or a cocktail of them. Okay. Questions on that? Okay. HMG-CoA reductase, by the way, happens to be the rate-limiting step in the cholesterol synthesis pathway. Okay, so that's why it becomes the major drug target for this condition. Now, the other aspect of treating this would be just simply lifestyle changes. Okay, so just reduce the amount of cholesterol intake for the individual. That can be, of course, done by changes in diet. There are actually resins that have been made that a person can ingest as well. Those resins tend to hold on to cholesterol in the small intestine, or absorb it, and then are removed through feces. The only way we get rid of cholesterol is through a bowel movement, so usually through bile salts. So that's another strategy in terms of reducing cholesterol in the body, is to just never let absorption occur. Okay. Now this does bring us to another topic, and that would just simply be diet. Familial hypocholesterolemia, those individuals are going to have high LDL concentrations, irregardless of the vigilance in which they maintain their diet and are on these drugs. Okay, so they just have it against them. However, diet can also affect this, right? So what you see here over on your right, once again, you have increased cholesterol intake. What you're going to have, of course, is lots of cholesterol extracellular that's going to get into the intracellular environment through receptor-mediated endocytosis. Of course, an individual without the familial hypocholesterolemia is going to take in significant amounts of that into their cells. Okay, that will lead to then significant amounts of oxysterols, and oxysterols, as we have said, is going to inhibit that process. It's the feedback inhibition, right? So if we come over here, okay, we can just say this over here on this side of this figure. We've talked about, of course, everything to the right, how LDL gets taken into these cells. What's on the left there is how these LDL receptors are made. As I mentioned on Monday, LDL receptors can be recycled from the endosome back to the plasma membrane, but there's a limited number of times that they can. Okay, so inhibition of the synthesis of LDL receptors will, of course, reduce the amount of LDLs coming into tissues, and that will then just put them into the bloodstream, again leading to the pathologies that we talked about at the end of Monday and the beginning of today. Okay. Now, what I want to go ahead and look at here real quickly is this. Okay, how do these oxysterols lead to the inhibition of LDL receptors? Okay, so that brings us to this slide, and what you are looking at here is the ER membrane. Okay, and in the ER membrane there are 3 proteins that are embedded in that membrane that are very important in terms of a cell's capacity to increase or decrease LDL receptor production. Okay, and those 3 proteins are listed here. The first one, we'll start here, SREBP, or it's got a really long name, sterol regulatory SR elements binding protein. I can just imagine the lab meeting that they all sat around. Well, what should we name this thing, that we discovered? I've got it. We'll name it sterol regulatory element binding protein. Excellent. And then we'll call it SREBP for short. Okay, so anyway, yeah, I don't even know how to pronounce it. So anyway, all right, so that's one of these factors. Now, the factor next to it, and again, another lab meeting, what do we call this? But this, SCAP, okay, that is SREBP, cleavage activating protein. Okay? SCAP. Or if you want to go in the long version, that would be sterile regulatory element binding protein cleavage activating protein. It's a mouthful. Okay? So that's SCAP. And then associated with those, finally, this last one would be INSIG. I'm not going to talk about I won't give you the abbreviation or the long term of that abbreviation, but INSIG. Okay? All right? And so those three, okay, when they are bound by oxysterols or sterols, so these would be the byproducts of some cholesterol metabolism, okay, those two proteins, SCAP and INSIG, can be bound by those things, okay, and when they are bound by those compounds, the three proteins that I've mentioned here are in a complex in the ER membrane, okay. Now, that complex holds all three proteins in the ER, so it prevents budding off from the ER into the Golgi apparatus. Okay. Now, if oxysterol and sterol concentrations go down, okay, for whatever reason in the intracellular environment, so these start to come off, concentration of these are getting lower in the ER. And because they're getting lower in the ER, then of course these proteins are, based off of their KDs for those, are losing the occupancy by those things. What will happen is this, the three proteins, okay, will dissociate. In fact, INSIG will dissociate from the other two proteins, and it will be marked for destruction by ubiquitin targeting pathways. Okay? It will get destroyed, and that will allow then these, okay, and they're kind of written over here, this would be a family of proteins called secretory proteins, or escort proteins. Okay? To bind SCAP and SREBP, that will allow these to go into the Golgi. So this would be step number 3. Okay? So INSIG gets destroyed. Escort proteins then bind SCAP and SREBP. Those then bud off the ER, go into the Golgi, and then the Golgi, they're exposed to protease. In fact, the SREBP is exposed to 2 proteases, okay, that cut here and here. Let's come all the way around here and release this tiny little green thing that then diffuses into the nucleus, or is transported to the nucleus. Okay? And that is the sterile regulatory element. Right? That's released once it gets into the Golgi. It will bind at promoters of a large number of genes that are involved in lipid synthesis, okay? Including the LDL receptor. Okay? So this will then lead to production of a large number of genes, including LDL receptors. Okay, so LDL are expressed. Oops, can't really read that. Okay, now again, come back to this. The reason that it's being expressed is because oxysterol and sterol concentrations are going down. Okay, so if those levels are rising in the cell, then they'll inhibit this pathway. They'll inhibit the production of LDL receptors. Okay. So again, for the person with FH, okay, or a person that is, high in has just a, fairly poor diet, high in cholesterol, okay, LDL receptor production will go down, and LDL then concentration in the circulatory system will go up. Okay, and that will lead to pathologies. Right? So of course, reduction of cholesterol is very important. Okay, intake of cholesterol. Questions on that? Okay. Now, that then does it, and we've talked about now the endogenous pathway, we've talked about the exogenous pathway. The last thing that we need to talk about, okay, and we've also talked about LDL, the bad cholesterol. The last thing we need to talk about is something called reverse cholesterol transport that has these HDLs. HDLs are the good guys, okay, high density lipoproteins. So let's go there, talk about this, and move on, okay, into other topics. Okay, so HDLs, again, high density lipoprotein. So we've got HDL here, lots of protein in this relative to the other components of it, fairly small amount of triglycerides. Okay, the composition, and as you'll see, of HDLs will change, and it will actually undergo what might be considered like maturation. Okay. As we look at this, okay, HDLs are very important because they have the capacity of removing cholesterol from circulation. Okay, they can actually remove cholesterol, as you will see, from LDLs and even VLDLs. Okay, well, actually, mostly LDLs. Okay, now let's talk a little bit about, first of all, how they are made. So let's put a cell over here. Okay, a plasma membrane, and we will just say that this is either a liver cell, or we could even see this happen in the small intestine. So the small intestine has the capacity to make these as well. The majority of it, like 85% of it, is going to be made, or 75 to 85% of it is going to be made in the liver. Okay, but a small amount of it can be made in the small intestine. Okay, so when we look at this, the very first thing that we have to recognize is that there is a protein, ApoA1. Okay, so when we look at these proteins, ApoA1 is one of them that's found in these. C2 is another one that's found. And then we'll just say others. Okay, but ApoA1 is actually excreted from the protein, from the cell as just the protein. I mean, no lipids attached to it as of yet. Okay, it's taken out of the cell. Right. These cells also come equipped with a protein, a membrane transport protein, okay, called ABCA1. Okay. There's a large number of what are called ABC transporters in the cell membrane. They're a huge family of transporters, and they are fairly complex in their overall structure and fairly complex in what they can move through the membrane. Okay? Generally speaking, though, they can move lipids through a membrane. Okay, so what will happen is that this can actually take phospholipids and cholesterol, not cholesterol esters, but cholesterol, and export these. Okay? Now, that ApoA1 that was exported will bind to this ABC transporter, and it will receive the phospholipids and the cholesterol, and that will make what is called a pre-HDL or a nascent HDL. And we'll look at that on our next slide. And it's just simply an apolipoprotein with phospholipids and cholesterol. Okay? That's all it is. Now let's go ahead and look at this on our next slide. So here's that ApoA1 that I was mentioning on the last slide. Okay. Again, it's this long, extended alpha helix. Okay, amphipathic, hydrophobic side and a hydrophilic side, right? And that can combine then, again, it will bind to ABCA1, that was that transport protein that I was talking about, and receive the phospholipids plus the cholesterol. Okay, and that will make this what I call pre-HDL or a nascent HDL. Okay, and you can see this. Hopefully you can see that. This is essentially this disc-like structure. It looks like a sandwich. It's got essentially, if you think about it, as a phospholipid bilayer in some senses, with a protein that wraps around that phospholipid bilayer twice. Okay, this alpha helical that wraps around it twice. The cholesterol, you can't really see where it is, but it's interdigitated in between the phospholipids, much like it would be in the membrane. Okay, and it gives this then some flexibility, okay, and mobility. And so that then is the production of HDLs. Okay? That's how they begin. Now those then hit the circulatory system, and here's what they will do. So we'll come right back to this slide here in just a second. Okay, but let me go right to here and kind of follow this pre-HDL or this nascent HDL and what it can do. Again, lots of details are being kind of just scrubbed over right here. But we'll get the general gist of this entire thing, okay? So we look at this here, okay? We've got essentially the liver, right? That's what's being shown here, 75%, okay, of that ApoA1. Like I said, it's 85 to 75% of the ApoA1 HDL production comes from the liver, okay? And then about 20% of it comes from the small intestine, SI, okay? Makes that particle that we just looked at, okay, ABCA1 is part of that. It hits the circulatory system, and when it hits the circulatory system, it will make its way to a number of different tissue types. Now this slide, okay, I took this actually from a review in Nature. This slide is showing really one aspect of what HDLs can do, but I think it's can be generalized from there. Now, first of all, let me do this. I probably should have done this right away. I'm going to go ahead and get rid of these boxes. The review is more based off of what oxidative stress will do to these different processes, okay, in what's called reverse cholesterol transport. So I'm going to go ahead and get rid of all those boxes. And just focus on reverse cholesterol transport. Okay, and that's what you're looking at here. This is a much better slide than what you find in your book. That's why I'm using it. Okay. All right, so these HDLs will then bind to a number of different ABC transporters on different cell types. Okay, they are showing in this a macrophage. We'll just put or some other cell. Macrophage is a very important aspect of this, right? Okay, because the macrophages, remember, are going to be the things that are absorbing excess LDLs out of the extracellular matrix surrounding the walls of the vascular system. Okay, other cells have these ABCA1 or ABCG1 transporters in them. Okay, the HDL will bind to these, and what they will do, stated right here, is that those ABC transporters will export cholesterol through the ABC transporter into the HDL particle. Okay, so what you're seeing here is this. Essentially, cholesterol comes out. Okay? And so these HDL particles will start to take cholesterol out of cells that have absorbed them. Okay, the macrophage being a very important aspect of that. So then, come back here. I'm going to jump between slides here. Okay? That's this process that I'm describing. Okay, these were the two factors I mentioned, ABCA1 and ABCG1, right? The absorption of essentially just straightforward cholesterol. Okay. Let me go ahead and write that down. There we go. Cholesterol. Now, HDLs, right? I mean, so they're becoming bigger. You can see that, right? They're starting to become, instead of discs, they're starting to become more spherical, more like what we've seen with the VLDLs and the LDLs and that kind of thing. They're still much more compact than those, but they're becoming more spherical in their nature. And it's important to understand this. it is absorbing cholesterol directly from these cell types, okay? However, there's this enzyme, LCAT, and that brings us to this slide here. LCAT stands for lecithin cholesterol acetyltransferase. So we've talked about ACAT, and now we're talking about LCAT, okay? LCAT will convert that cholesterol that's being absorbed by the HDL into a cholesterol ester. Okay, so it will take this LCAT protein, will take from phosphatidylcholine, it will take one of those acyl groups off. Remember, this is a phospholipid, so it's got two acyl groups and a phosphate head group. Okay, it will take that off and it will put it onto cholesterol, making a cholesterol ester. And it then will make the byproduct lysolecithin, which is a strong detergent that remains in the HDL particle. Okay. And that's all done by this LCAT enzyme. Now you might ask yourself, well, what's the importance of that? I mean, that's great. It's converting cholesterol to cholesterol esters. Why? Just so I have to know another enzyme name? Exactly. No, it's actually more important than that. So cholesterol, as it sits in this form right here, when it gets into these lipoprotein particles, it will, as I said, find its way into the membrane component of that, the phospholipids. Okay? And at certain levels, that's good, right? It'll cause some fluidity in that. However, if it starts to get too concentrated in that, it will disrupt the structural integrity of the particle and essentially the phospholipid surroundings. And so if it disrupts that structural integrity, these particles can actually start to fall apart in your bloodstream. So what needs to happen to that cholesterol as it gets absorbed by HDL is it needs to be converted to cholesterol esters. When they're converted to cholesterol esters, this hydroxyl group that would allow it to interact with the polar head groups of the phospholipids, that has now an acyl group attached to it, a hydrocarbon chain. It becomes very hydrophobic and it moves into the interior of the HDL particle, right? And so then it starts to look like this, okay, and it maintains the structural integrity of the HDL particle. Okay, so LCAT is actually fairly important so that cholesterol esters can be produced and cholesterol can be put into the interior of these HDL particles. Okay. Now, once we get to there, okay, so we have this HDL, and we're not going to go any further on that slide. I mean, there's different forms of HDL, but once we get to this HDL that's absorbed cholesterol from either a macrophage or some other tissue, it has had it converted to cholesterol esters, it will start to make its way back, okay, and this is where we get this idea of reverse cholesterol transport. It will start to make its way back, okay, and we're going to skip some steps here. For instance, we'll not talk about that. It'll make its way back to the liver and it will bind to something called SRB1. Okay? HDL binds SRB1 and transports cholesterol esters into the cell, and it's actually, I guess I should say, I didn't mention this, but the liver cell. Okay. SRB1 is concentrated, that receptor is concentrated on liver tissue. Okay. HDLs will come back there and they will start to export cholesterol esters into that cell. Now, it's not endocytosis. Okay, so it's just straight movement of the cholesterol ester out of the HDL into the liver. Those cholesterol esters will then hopefully, much of them, as they come in here, CE, will be converted to cholesterol and then to bile and taken out of the, they actually have that on this slide. Cholesterol will be converted to bile and then excretion into the colon. Okay. And then you can remove it. Okay. So you notice what's happening here. This is why HDLs are called the good cholesterol, because they start with really no cholesterol in them. They go to the tissues, they grab the cholesterol out of them, convert it to cholesterol esters, take it back to the liver, put it into the liver, and hopefully the liver gets rid of it in the bile. Okay, now some of it, of course, is going to come back. They're going to make VLDLs and that kind of thing. Right. Question, yes?
So are there any disorders where the liver actually doesn't properly deal with cholesterol? Like, we've heard about hypercholesterolemia.
Yeah, like once it's in the liver, not processing it correctly, that kind of thing. I'm sure there are. I don't know any off the top of my head. But yes, I mean, there are a number of ways to die. So I don't know of any, but there are a lot of steps in these processes that could be potentially monkeyed with that can lead to disorders. Well, I wouldn't know. I mean, without knowing what step and what is the accumulation of what, metabolite would cause what pathology, it would be different. And what pathology would that do to like circulating LDLs? I don't know. Yeah.
So HDLs are only good because they're removing LDLs from your body or from your liver or small intestine?
Yeah, they're not really. Yeah. So the question is, the HDLs are only good because they're really removing LDLs from your body. Not really. They're not really removing LDLs. They're removing the cholesterol that made it into the macrophages from the LDLs. Okay. So they're considered good cholesterol. Okay, because they have the capacity to take cholesterol from essentially the systemic system, bring it to the liver, with the hopes that the liver will then get rid of it. Okay.
So if your HDL levels are low, is that not a problem if your LDL levels are also low?
Well, so the question is, if your HDLs are low, is that not a problem as long as your LDLs are low? It can still be problematic in the sense that then you can still have increased cholesterol accumulation, okay, in the systemic system, okay, because there's nothing to remove it. So when you start to talk to a physician or health providers, they'll say, well, you want your bad cholesterol LDL below 200 milligrams per deciliter, but that assumes a certain level of HDL as well. Okay, so and I can't remember what the suggested level of HDL would be in the bloodstream. There has to be a ratio with that. If that ratio is off, meaning that HDL gets lower than the suggested ratio, then that even lower than suggested LDLs can still cause a problem, if that makes sense. Okay, so they're looking for the appropriate ratio. The higher the HDL concentration, the better. Okay, so I'm sure within limits. I imagine, I don't think there are any, but I imagine there could be pathologies with too much HDL, but I don't know what they are. Other questions? Yeah.
Are HDLs able to break up or absorb some of the cholesterol from the plaques that form after the macrophages reach toxic levels of cholesterol, or is it only able to take that?
Yeah, so it would have to be, so the question, if you didn't hear it, is so these plaques that form that have the cholesterol in them, can the HDL take from those plaques? It's possible that they could. Although I would say this, it's not going to be enough to reduce the size of the plaque. The plaque is insoluble mess, and so the accessibility of the cholesterol and its ability to get into the HDL will be significantly less. Not only that, but so this is something I'm not clear on, okay, and that makes it hard for me to answer that question. Right, the ABC transporters have to bind the HDL, right? And certainly they are transporting something out, so there's a mechanistic aspect to transporting cholesterol out of the cell. But are they also then having some mechanism that either we don't know about or I don't know about it for sure, that allows then cholesterol to be put into those HDLs, or is it just a diffusion process just in proximity? If it truly is just diffusion process, right? I mean, HDL is just there and it diffuses into it. Then you could see that maybe the HDL could absorb cholesterol from, something else, from the plaques. Though it wouldn't be targeted to it, right? I mean, why would it associate with those, right? So, so that would be the other aspect of it. Yes?
So when your HDL and when your cholesterol levels are checked like in a blood test, are they not actually testing for the cholesterol but for the HDL?
That's right.
The concentration that needs to be for the
Yeah. Yeah, I mean, you say, I mean, we talk in, I mean, colloquial terms, I'm going to have my cholesterol checked or my cholesterol is high. What we really mean by that, though, is you're checking LDL and HDLs. You're not actually checking free-floating cholesterol in your bloodstream. You don't have very much of that. But they're checking for levels of LDLs and HDLs. And I don't know, honestly, I mean, I don't know the analytical biochemistry behind that. I would imagine that the blood screening has to do with targeting the proteins found in the lipoprotein particles that are unique to those particular HDLs and LDLs. Yeah. So it's not really, I mean, we call it cholesterol, but it's these lipoprotein particles. Other questions? All right, we haven't quite finished with this slide. There are some other things on here, and you say, great. I was hoping you were going to say that. So let's look at this. I do want to say one more thing in regards to this and talk about one more protein. Okay. So HDLs, interestingly, can also interact with VLDLs in circulation. Okay. And the HDLs can, interestingly, transfer cholesterol esters to VLDLs via this protein called CETP, cholesterol ester transferring protein. So CETP, cholesterol ester transferring protein, okay, can actually mediate exchange of cholesterol esters from HDLs into VLDLs. It will also exchange in that process triglycerides, okay, that then can be unloaded by HDLs into the liver. Okay. Now, that process, when we look at it, then that puts more cholesterol esters in VLDLs, which are packed with, remember, triglycerides. These are made at the beginning of the endogenous pathway and excreted by the liver. Okay, these will undergo the transformations that we talked about to LDLs, with the hopes that then these get back and are absorbed by LDL receptors by the liver, and then the cholesterol can be excreted by bile. Okay. Right. Of course, problems could arise where it could go back to an accumulation of LDLs, and everything that HDLs are trying to do, okay, is being counteracted by overaccumulation of LDLs. Okay, but that does happen. I mean, we have observed it, right? That cholesterol is removed, or cholesterol esters are removed from HDLs and put into LDLs by this cholesterol ester transferring protein. Okay, so. Now that's reverse cholesterol transport. Okay, any questions on that? We should finish right there. I mean, that's a perfect place to stop. That's the end of the reverse cholesterol transport. Okay, so Friday hopefully we'll come and we'll finish out this set of slides, and then we'll be able to start lecture 11 on Monday.